Yeah.
It's all arbitrary.
My stat professor always used real world stuff. We did baseball statistics. We did analyzing scientific papers. We did all of kind of that. That was really fun. And he gave us big group projects that we had to work out together. 5 of us graduate students, we all had to work it out together. So I ended up liking statistics, but I can understand why you wouldn't. Okay, so hopefully we're going to finish this set of notes today. And yes, I am recording. You're not going to have a quiz on Tuesday, but you will have one on Thursday. Okay? So before we get into the new material, let's review to see if the enzyme stuff stuck. Okay? So I'm going to give you a couple of scenarios, and what I want you to do is tell me whether the scenario would increase or decrease the chemical reaction rate. You with me? Okay. First, what increasing substrate concentration would tend to do what to the reaction rate for an enzyme catalyzed reaction? Increase, yes? Until what? Until you hit saturation, yes. Okay, so now let's go to saturation. The only way to increase the saturation rate is by increasing what?
Substrates.
No, not substrate, that won't do it. You have to increase the concentration of the enzyme. Does that make sense? Increase the concentration of the enzyme. Okay, so now let's go to the two that people get confused, okay? And the concepts of substrate specificity and enzyme substrate affinity. And let's see if you guys can separate those out for me. So substrate specificity first. Can anybody raise their hand and give me a little synopsis of what substrate specificity means? Go ahead.
So there's a certain substrate that bind more easily
Yes.
and quickly than there's others that might work, but it'll just take longer and be a more energy inefficient process.
Okay. So if the substrate fits better, what does that predict about the chemical reaction rate?
Faster.
It's going to be faster, right? Substrate specificity increases, right, the rate of the chemical reaction. Okay, so now let's do the other one. Enzyme substrate affinity. Enzyme substrate affinity is Michael and I are both humans, and we have similar enzymes, otherwise we wouldn't still be alive. So I'm going to dump the same identical substrate into us, and enzyme substrate affinity says that his enzyme might fit the substrate better than mine. And what does that predict about his rate of chemical reaction versus mine? Faster, exactly, right. Okay, so that enzyme substrate affinity has to do with, remember, enzyme substrate affinity has to do with multiple possible what? Enzymes. And substrate specificity has to do with multiple possible substrates. Exactly. Does that make sense? Okay, you got it? Okay, and as we mentioned last time, what's actually true in the human body is that I probably have both going on at the same time. Because for every enzyme, I have potentially 2 different forms because I've got 2 parents, right? 2 different forms of any enzyme, right? And also a whole bunch of substrates. Does that make sense to you guys? Okay, so those are those 2. By the way, there's a little hint in there as to which one's which. Only one of them has the word enzyme in it, yeah? Enzyme substrate affinity has to do with multiple possible enzymes. The other one just has the word substrate in it, so that has to do with multiple possible substrates. Okay, so the last thing that we did last time was product concentration. What was the prediction about how product concentration would affect the rate of enzyme catalyzed reactions in a metabolic pathway? So what does product concentration often do?
Turns on or off the production of that certain product.
Yeah.
Based off negative feedback.
Yeah, and so as product concentration increases, what does it tend to do to the metabolic pathway?
Temporarily stop it.
Temporarily turn it off, right? Okay, and then the product concentration will go down, right? And as the product concentration goes down, then more of the enzymes are on, right? Just because of the likelihood of the more product you have, the more likely it is to find that binding site or regulatory site on the enzyme, right? That makes sense? Okay, so that's product concentration. And that leads us directly to the thing that we're going to cover now, which is regulation of enzyme activity. What we just looked at was an example of that, but we didn't call it that because we weren't quite ready for this terminology yet. So what we just did, yes, with the product concentration thing, was bind at a non-active site, right? Not at the active site, at another site called the regulatory site. Something. Yeah, in that instance, it was the product, but this is global. This is bigger, okay? This generally is just called a modulator, but that's just a general term. It means something that binds at a regulatory site. And notice what the binding at the regulatory site does. It temporarily and reversibly changes the shape of the active site, yeah? Now, how is it temporary and reversible? Well, because generally modulators, or the product that we were talking about before, binding to the regulatory site is hydrogen bonding and or ionic bonding, which is easy to form, easy to break, easy to form, easy to break. Does that make sense? Okay, so it's relatively loose and reversible. Yeah, so it's not as if this modulator binds and then stays there forever. It binds and falls off, but if there's a high concentration of modulator, then another one might find it, right? Does that make sense to you guys? Okay, so this is called allosteric regulation, and allosteric regulation, modulator binds and alters the enzyme shape by binding at a regulatory site such that the shape of the active site is changed temporarily. So what does this have to do with regular everyday life? Well, some things that you know that you need in your body, but you often don't know why you need in your body, are used in this way. And so very, very common modulators in the body are needed in trace quantities, but you never really thought about why you need them. So for instance, a lot of times people, of course, know why you need carbohydrates, why you need proteins, why you need these big macromolecules. You're going to use them primarily for catabolic processes, right? And then sometimes you'll use the components for anabolic processes again, right? But you never really think about why you need copper, right? Or why you need vitamin C, okay? So quite often what you do is you need those as modulators in a biochemical pathway to turn things on and turn things off. Yeah? Does that make sense? So that's why you need them in microgram quantities or milligram quantities and not gram quantities like you do, for instance, carbohydrates. Does that make sense? So these are, they're called cofactors or coenzymes. And what they are going to do, these cofactors or coenzymes will temporarily change the activity in an enzyme that exists. So examples of modulators, so magnesium, zinc, copper, those are minerals. And then you also have vitamins. You may or may not know the difference between a vitamin and a mineral. They're both needed in trace amounts, not large amounts, so you're not using them for catabolic processes. And usually what happens is they are needed in parts of a biochemical pathway. And you might just, for instance, need at the end of a biochemical pathway a little copper to include in this chemical reaction. And so see if this makes sense to you. I've got a 32-step biochemical pathway that will, at the end, need trace quantities of copper to finish, right? Can you see that it might make sense to use copper as a modulator several places in the pathway? It's basically checking to make sure that you have it before you exert all of that energy, all 32 steps. Does that make sense? So you would use these both as something final in the product pathway, but then also as an indication that you should continue at points 3, 6, and 17. Does that make sense? So these are cofactors or coenzymes. With that in mind, can you see why sometimes when people are deficient in vitamins and minerals, it can impact their metabolism? Can you see that? Most of us are probably not. We live a relatively privileged life in the Western world, and we probably don't need a multivitamin or mineral. Primarily when you take one, with most of us, you just pee almost all of it out. But if, for instance, you are a vegan and you know that you need certain vitamins that are difficult to get from plant products, you can impact your metabolism positively by making sure that you have adequate amounts of those trace nutrients, okay? So vitamins and minerals are often used as cofactors or coenzymes to regulate the activity of existing enzymes via allosteric regulation. And allosteric just means that temporary shape change with hydrogen bonds and ionic bonds loosely and reversibly binding to a regulatory site. Okay, so please look at what you're trying to accomplish and then hold that in your mind before we get to the next ones. Notice that what we are doing is changing the shape of the active site. Okay, so before we go to the next thing, let me just show you how that would affect the activity of the enzyme. Here's a common enzyme, right? You've got the chemical reaction rate, and it's going to increase as the substrate concentration increases until you hit saturation, yeah? And so you have this typical curve for how an enzyme would actually, this is an enzyme catalyzed reaction. But what if it's an allosteric enzyme that needs to be activated? Do you see that you are actually going to have a slower upsweep because you're waiting to activate it? That makes sense? So it kind of like indicates to you that that one is one that's probably not on by default, right? It needs to be activated. Okay, so one more time, notice the shape of the active site. And now I'm going to tell you that there is another really common way of activating enzymes. And this one is called covalent regulation. And so let's just introduce you to it, and then we'll get to the details. What I want you to see is that the outcome is the same. Do you see active site changing the shape of the active site? The process is going to be different, and it's going to be a more permanent change, and permanent with an asterisk because you can turn it off, but it's not going to fall off on its own, okay? So covalent bonds compared to, for instance, ionic or hydrogen bonds, stronger or weaker? Much stronger and a little harder to form, yes? Okay, so what we do with covalent regulation is we are going to change the activity of the enzyme, right, by changing the shape of the active site, and it's going to stay changed until we purposely undo it, okay? So with covalent regulation, what I'm going to do is it's actually not called the regulatory site when you use covalent regulation. What I'm going to do is I'm going to take a chemical group like a phosphate, yeah? And I'm going to slap it onto this enzyme, yeah? And now I've got it stuck with a covalent bond. And that would be, for instance, how you activated it. And it would stay active until you broke that covalent bond, yeah? Okay, so the most common way to do this is by using a phosphate. It's not exclusive. You can also use a methyl group or something else. But I know you've probably run into the phosphate part before. Okay. So phosphates are relatively easy to access because, of course, you can always take them from ATP, right? Yeah? Adenosine triphosphate, you can pop a phosphate off. Okay, so stick with me for a second. So what's going to happen is in order to do covalent regulation, which really accomplishes the same thing as allosteric regulation, you're going to need two friends, yeah? In order to manipulate this enzyme, you're going to need an enzyme that can pop it on and an enzyme that can pop it off. If this group were phosphate, let's say it is, then the enzymes you would use, you already know the name of. Do you know the name of an enzyme that takes a phosphate and puts it onto something? Kinase. And do you know the name of an enzyme that takes that same phosphate and pops it off? Phosphatase. So you will see that this is a really common way of regulating enzyme activity. So let's go back a little bit and talk about why the hell you would want to regulate enzyme activity. Okay, so we talked about this before, but just to cement it in your head, why would I, for instance, not just make an enzyme when I need it and not make it when I didn't need it? Right? It's not necessarily energy, it's time, right? So if I need an enzyme at certain points and I don't need it at other points, do I really want to wait until I can go all the way through protein synthesis before I have that enzyme, right? So what happens is when I need enzymes sometimes and not others, I have the capacity to have the enzyme pre-made already there, but I can manipulate it by turning it on or turning it off. This mechanism is turning it on and the damn thing stays on until you turn it off. The other one is just like a little negative feedback inhibition based on concentration, right? It's just going to change the activity whenever it encounters a modulator and not change it when it doesn't, right? Does that make sense? So these are just two ways of regulating enzyme activity. They accomplish the same thing, but one of them is more permanent than the other, and permanent with an asterisk because you can turn it on and it stays on until you turn it off. So this is called covalent regulation. And kinases and phosphatases are really famous for doing covalent regulation. Okay, so now we're ready to move away from enzymes for a little bit and start talking about metabolic pathways. Okay, so metabolic pathways are, remember, a series of enzyme-catalyzed chemical reactions, right, before you get to the product. So you may have 32, you may have 75. So I've got all of these reactants down at this end, right? Series of enzyme-catalyzed reactions until I get to the product, yes? So that's a metabolic pathway. And arguably in humans, the most important metabolic pathway is cellular respiration. And cellular respiration is really something that this time, if you're anything like me, you've learned it like 7 times and you never remember it because it's not relevant. So my objective today is going to be to make it so relevant that you'll have difficulty forgetting it. Okay? So cellular respiration in humans, animals, everything really that does it, is the process by which you take big organic molecules and break them down to smaller organic molecules and give ATP off as a byproduct, right? And so what's the big picture? Why the hell do you need ATP? Yeah, it's the currency. And in the absence of ATP, what would not happen physiologically? You couldn't maintain homeostasis, right? So this is intimately related to the process of physiology in 100 different ways, right? The big picture is, of course, you can't maintain homeostasis. You wouldn't be able to stay an organized human in the absence of ATP, not even over the course of a day. So cellular respiration is going to be the process that we're talking about. But let's just back up just a minute and say cellular respiration is badass, absolutely necessary. I hated it too. I tried to resist learning it for as long as possible until somebody finally convinced me that it was relevant to anything. But you can do cellular respiration, and all animals can do cellular respiration, but you are not as cool as a plant because plants can do the other process too. Photosynthesis is the process of taking sunlight and organic molecules and making the thing that you can burn so they can make glucose out of it. That is amazing because then they can also take the glucose and do cellular respiration and make ATP out of it. So let's go back for just a minute and talk about what we are doing to our planet, right? And the fact that we are really, really depleting natural resources and we're causing a lot of pollution. People are constantly saying that we are destroying the planet, and there's an argument for that. But the important thing is not that we are destroying the planet, but we are destroying the planet for us. If you, however, could make food out of sunlight, you're probably going to survive. But we can't. So the autotrophs, after we leave, yes, we may lose all of the big trees, but will we probably lose the photosynthetic organisms? No, they're basically going to do some pest removal, give it a few millennia, and then they will probably bounce back, and that's probably the last time you'll see humans, though. So we cannot do both of those processes. We can only do one of those processes, and we are dependent on them for the other process. And it's interesting to think about as we are trying to kill them all. Okay, so let's talk about cellular respiration. So enzymes are necessary for each reaction in the pathway. And you already know, because you've learned before, that cellular respiration is basically 3 groups of chemical reactions. There's glycolysis, yeah? Okay. There's Krebs and the linking reaction, Krebs cycle. And then there's the last set, which is either called electron transport or oxidative phosphorylation. Okay? So memorization is not what you're going to go for today. You're going to go for understanding to the point that you could explain this to a really smart 10-year-old. If you can't do that, you don't actually understand it, okay? I could explain almost anything to my 12-year-old child related to physiology because he is freaking brilliant, and all I have to do is get the metaphor right, and he'll get it, okay? So that's your objective today. So enzymes are necessary for each step in the reaction. So here is how, if you were taking biochemistry, has anybody taken biochem yet? Okay, so if you're taking biochemistry, when you learn glycolysis, you will learn the whole damn thing and you have to draw it and know all of the enzymes, but I'm not going to do that to you. So you have to learn its relevance to human physiology. So we really have to put it together as a story that's relevant to human physiology. What glycolysis is the first step of cellular respiration, right? And it's really 10 enzyme catalyzed reactions, okay? And really what you're doing, big picture in glycolysis, is you're taking a big sugar, generally, not always, and you are going to break it down to smaller components. So glycolysis is only the first step of cellular respiration. So let's simplify cellular respiration as much as possible. And I don't need you to balance this equation. I just need you to look at the big picture. The big picture of cellular respiration is that you start with a big organic molecule, and this one happens to be glucose. But let's acknowledge that you can burn things that are not glucose. You can actually use them. Okay, and then if you want to get all out of glucose you can, you need to have oxygen. And then this arrow actually represents a whole bunch of chemical reactions, right? Because I'm not going to go through all of them. This is what goes in, and ultimately the summary of what comes out of cellular respiration is carbon dioxide, water. This is called metabolic water. You don't really have to worry about it though, because it just mixes with all the other water in your body. And then the point of it all is because this makes what? ATP, yes. So this is the point of it all. Okay, so my objective today is going to be to convince you that this is worth learning. And I'm going to start by telling you that it absolutely is worth learning, because this is the entire reason that you breathe. Isn't it? And that couldn't be more relevant to physiology. Why do you breathe? Well, because you can't break this down very well in the absence of oxygen, and because you must get rid of this byproduct. What's one of the reasons that you've already learned that you must get rid of this byproduct? Yeah, if you don't get rid of carbon dioxide, it will affect your pH. If you affect your pH, your proteins stop functioning. So it is called cellular respiration for a reason, because at the cellular level, it's oxygen in and carbon dioxide out, right? So it's the entire reason you breathe. And I would also argue that it's the entire reason you have a digestive system. Yes? Because you need to get in large nutrient molecules that you can break down. Glucose is not the only one that you can do. There are others, but you have to take them in via your digestive system. It's also almost the entire reason that you have a cardiovascular system, because you have to get those from the sites at which you take them in, respiratory system, oxygen, digestive system, nutrients, to the sites at which you use them, which is every damn place in the body. And diffusion does not work very well for an adult human, okay? So this is fundamental to understanding physiology, even though it's not everybody's favorite thing to do. So here's how we're going to cover it today. What we are going to do is we are going to keep track of where all of these reactants are used, whether they're used in glycolysis, Krebs, or oxphos, yeah? And where all of these products are generated, whether they're generated in glycolysis, Krebs, or oxphos, right? And then ultimately what you're going to do is to try to come full circle at the end here so that you can put this back into the content of being relevant to human physiology, okay? All right, so here we go. Glycolysis, the name implies what it does. Glycolysis is glycolysis, the breakdown of sugars, okay? And it doesn't have to be glucose, but glucose is the one that everybody memorizes. Why? Because every cell in your body loves glucose, even the brain. It's not the only thing they can burn. You can burn almost any simple sugar, but this is probably the one that you have memorized. So carbohydrates are going to start their breakdown on glycolysis. Let's look at a simplified version of cellular respiration. It doesn't look very simple, but it actually is. Okay, so here's glycolysis. Instead of the 10 reactions that make you want to cry, they're like, here's a purple box. It's a pleasant purple box. Don't worry about the purple box. Okay, so glucose goes into glycolysis. What comes out the other end is going to be 3 carbon molecules called pyruvate or pyruvic acid. Okay, and importantly, you get a little bit of ATP, right? So that's the sort of summary of what happens in glucose. So let's go back to your notes, and then we'll fill in this. So glycolysis is interesting because glycolysis, although you've memorized in your head that cellular respiration happens in the mitochondria, that's only truthy. It's not true, because glycolysis actually happens in the cytoplasm or cytosol. And interestingly, glycolysis is older than animals. Glycolysis happens in bacteria. It's interestingly also older than high concentrations of oxygen on the planet. So it doesn't even require oxygen. So in the absence of oxygen, whether you're a bacteria or a human who is working out faster than your muscles can get oxygen, you can still do glycolysis, right? So glycolysis is a really, really old form of gathering energy, and all cells can do glycolysis. All? I bet all. So this is where carbohydrates start the breakdown, and it occurs in the cytoplasm or cytosol. It doesn't require oxygen, so it can and does occur in anaerobic conditions, whether the anaerobic condition is an anaerobic bacterium or your skeletal muscle that you're working out faster than you're actually feeding it with oxygen from the bloodstream. So you can still do that. Now, if oxygen is present, the big part about this is that if oxygen is present, you can keep going after this. And that's important because we will see that although glycolysis is a good start, it doesn't get you much ATP at all. So let's talk about how much it's got. So if oxygen is present, you will continue after glycolysis. If oxygen is not present, you get very little ATP formed, and in some of your tissues, you will start to form lactic acid as a byproduct. Yeah? In you, if you continue to force your tissues, skeletal muscle for instance, because not all tissues can do this, if you force skeletal muscle to do glycolysis over and over again, because you don't, for instance, have oxygen, then you will build lactic acid as a byproduct, which does not cause muscle soreness, but does cause muscle fatigue. Okay? People have been teaching that it causes muscle soreness for decades, but it doesn't. It causes muscle fatigue. But other tissues can't do that. So your brain cannot do anaerobic respiration, generate lactic acid as a byproduct. It just freaking dies if you cut off oxygen. It's like, screw it, can't do it. Okay, so let's keep track. How much ATP is formed from a molecule of glucose one time through glycolysis? So what we're going to do here with each one of these is keep track of where they're used or where they're generated. Okay. In glycolysis, did I use glucose? Yeah, of course. I used glucose in glycolysis. Did I use oxygen? No, I didn't use oxygen. Did you see me generating any carbon dioxide? No, all the things that you generate are over on this side. Did you generate any carbon dioxide? No. And you shouldn't have, right? Because you would have needed oxygen to generate carbon dioxide. Yes? You don't generate carbon dioxide. Did you see me generating any metabolic water? No? How about ATP? Yeah, you generate 2 ATPs net. Basically, it takes 2 to run it, but you generate 4. So we'll just keep track of the net. So you generate 2 from glycolysis. Got it? Okay. Now, why the hell do you care about glycolysis when it only generates 2 ATPs? Well, it also generates pyruvate or pyruvic acid, which is 2 3-carbon molecules. And those are important because those can go into the linking reaction in Krebs, right? And Krebs isn't very impressive either, but Krebs will get you to oxphos, which is when all the action happens, okay? So what's going to happen here is these 2 right here are going to start the next part of cellular respiration. And it's the products of glycolysis that will go into this. And the products of glycolysis go through this linking reaction. This, by the way, is the linking reaction. I don't care if you remember what the linking reaction specifically does, because I usually lump it in with Krebs. Okay? So the linking reaction takes pyruvate and makes it into acetyl coenzyme A. Coenzyme, we just talked about coenzymes. And then that can go through Krebs. So let's look at what happens in Krebs and why the heck you care about Krebs. Krebs and the linking reaction. Okay, so if you are a carbohydrate and you started here, if you were glucose, and you got to here, and there was no oxygen, that would be as far as you could go. If there is oxygen, however, you can go through the linking reaction in Krebs. You with me? Okay, so carbohydrates will continue their breakdown after glycolysis by going through Krebs. Krebs occurs in the mitochondria. Oxidative phosphorylation occurs in the mitochondria. Everything that occurs in the mitochondria requires oxygen. Okay? So, what I'm going to get is from Krebs, the important thing is I'm going to get the stuff that I could stick into oxphos, and then potentially I might get a little bit of ATP. So, from Krebs, I am going to generate not an impressive amount of ATP. People argue a little bit about the numbers, but it's usually either 0 ATPs or 2 ATPs, depending on which way you go through Krebs, okay? Either way, it's not a very impressive amount. So let's go back to here for just a second. Did I use any more actual glucose? No. Did I use oxygen? Yeah. Did I generate any carbon dioxide? Yeah. All of the carbon dioxide is generated in the linking reaction in Krebs. Do you see it? Okay, so that's carbon dioxide. Did I generate any metabolic water? No, metabolic water comes later. Did I generate any ATP? Potentially 0 to 2 from Krebs. But please note that it's still pretty damn unimpressive, yes? As far as the quantity of ATP that is generated. So the quantity of ATP is still not very much per glucose molecule. That's because where I was trying to get is right here. Another coenzyme, guys. Coenzyme. I just taught you guys about coenzymes. Coenzyme 2H. So why the hell do you care about that? Because now what I can do is I can do oxidative phosphorylation or electron transport. Oxidative phosphorylation, electron transport is really freaking complicated. You've probably learned it before. You probably hated it as much as I did. Basically, I'm not going to test you on this. You keep moving electrons until eventually they all end up on oxygen and you generate a whole bunch of ATP in the process. How much? Usually about 34 ATPs per glucose molecule. So that's when it actually gets impressive. So let's not look at it this way because nobody wants to talk about NAD, NADH, NAD, what the hell. So let's go back and look at it this way and say, okay, I successfully got through glycolysis. I got through Krebs. I still have oxygen present. If I do, I will go through oxidative phosphorylation, and oxidative phosphorylation is going to get me the money, which is 34 ATPs per glucose molecule. So if I'm a carb and I still have oxygen, I'm going to continue my breakdown there. Okay, it's going to use the collective products of glycolysis and then Krebs. Yeah, it's going to occur in the mitochondria, and since it occurs in the mitochondria, it requires oxygen, exactly. And it's going to produce metabolic water as a byproduct and 34 ATPs, which is the big deal there, okay? So one more time. I'm going to go back through one more time. Did I use any more actual glucose? No. Did I use oxygen? Yes. Yeah, I used oxygen. Did I generate any more carbon dioxide? Did I generate any more carbon dioxide in oxphos? No. Did I generate metabolic water? Yeah. And the big deal is that I generated 34 in oxphos. So, how many ATPs, the range of ATPs, do you generate with a single molecule of glucose in the presence of oxygen? 36 to 38. Good? Okay. In the absence of oxygen, how many do you generate? 2, right? So oxygen is pretty damn important for this process if you want to keep yourself organized. FYI, textbooks disagree on this number just a little bit because there's a few different ways that you can go through this, but it's always going to be in the 30s. Yeah? Okay, so let's go back full circle and talk about this big picture so that it's not just rote memorization, okay? So what you are going to need to do on your first test is a couple of things related to this. First of all, you are going to have to do this. This is the unbalanced summary equation of aerobic cellular respiration, yes? Don't balance it unless you know how to balance it, because if you balance it wrong, I will count it wrong, okay? So this is the unbalanced summary equation, okay? You need to know, of course, where each thing is used or generated, just like we have here, right? And the only thing that you need to keep track of numbers for is ATP, right? Okay. And then one of the things that I'm going to challenge you to think about is I'm going to challenge you to relate cellular respiration to human physiology, right, in 3 sentences or less. And the crux of it is probably going to be which of these molecules. Yeah, it's going to be ATP. So as we are going through the rest of the lectures before you get to your first test, you are going to say, bing, ATP, and you're going to say, okay, I'm going to put together 3 different examples of how cellular respiration is necessary for instance, running sodium-potassium pumps. You can use homeostasis. You can use words like this. So what I want to do is take this from being irrelevant, right, to something that you can put directly related to homeostasis, to human physiology, okay? 3 sentences or fewer. Good? And I'm not going to give you the answer because all you have to do is listen for the rest of the time, and then you start writing 3 sentences as you go through. Does that make sense? Okay. So, you got it? Questions? Go ahead.
So you're basically, you're going to want us to write something very similar to this. Or just
I want you to write this.
Yeah.
Yes?
Yeah.
But then I'm also going to ask, and the question is going to almost be verbatim that on the first test. It's going to be a big blank, and I'm going to say, in 3 sentences or fewer, relate aerobic cellular respiration to human physiology. And if you give me substantive examples, then you get full credit. What do you think? How much credit do you think I'm going to give you if you say you would die without it? No, because you have literally known that since 7th grade. Give me something that's actually putting it together with human physiology. And so what should you do? You should listen as we go through and start using ATP and say, that's a fantastic example of something that wouldn't happen in the absence of cellular respiration. Yeah? Okay, so we're going to try to make this relevant and try to put it all together with human physiology. Everybody okay? All right, it's not that hard, right? Okay, so now let's talk about stuff that you probably didn't have, maybe, I don't know, in 1610, and start talking about the fact that although of course you can burn glucose, and you've probably memorized some variation of this equation before, and by the way, you only have to memorize 2 chemical equations for my class throughout the semester, and you already know both of them. One of them was the relationship between carbon dioxide and pH, which we will use all damn semester. And then there's this one, which you probably should have known already, right? And we're never going to stop using it, ever. Okay, so now what we are going to do is we are going to talk about the fact that, of course, everybody's known for a long time that you can burn glucose to get ATP, but that's not the only carb you can burn. And in addition, you don't just burn carbs to get ATP. You can also break down proteins to get ATP and break down fats to get ATP. Now, nobody's ever made you memorize those equations for instance, the equation for lipolysis to get, and I'm not going to make you memorize the equations, but I'm going to make you put it together so that you can see the relationship of burning this nutrient to burning other nutrients, and then what you would do in this situation. So first, we're going to talk about carbohydrate metabolism globally. So let's back up just a little bit and say that generally speaking, in order to go into glycolysis, you are either going to need glucose or another simple sugar. But of course, we don't just eat simple sugars, right? We eat complex carbohydrates, right? We also make complex carbohydrates. So what would I need to do if I ate a complex carbohydrate like whole wheat pasta, right, in order to do glycolysis with it? Well, I'd have to start breaking it down to simpler sugars, right? Ultimately, glucose or something like it, right? So let's talk about how that is related to having a body like you. So I take in a complex carb, and I am going to start breaking it down. The breakdown process starts in my mouth with salivary amylase. You guys probably heard of salivary amylase before. Salivary amylase will break down carbohydrates in your mouth, which is why, for instance, if you get a cracker stuck in your teeth, it ain't gonna be there 45 minutes later. If you get a piece of steak stuck in your teeth, it could be there 3 years later, right? Because proteins do not break down with amylase. Does that make sense to you? Okay, you gotta get that crap out of there or else you're gonna get some gingivitis. Okay, so you're gonna break it down. So amylase starts here. I also have pancreatic amylase, which will break down complex carbohydrates too, right? But it starts in the mouth. So you're gonna break down complex carbs to smaller sugars, like disaccharides or monosaccharides like glucose. And then those are the ones that can enter glycolysis. Now, note to self, again, even though we're only showing glucose here, it's very similar for most. I mean, the quantity might not be exactly the same, right? But it's very similar for most simple sugars. Good? Okay, everybody good with that? So you can burn other things is what I'm trying to say. Now let's put this back together with human physiology globally for a second before we actually get into storage and that kind of stuff. So what happens in a person who, for instance, is an untreated diabetic who takes in sugars or even complex carbohydrates, right? An untreated type 1 diabetic. Can this process occur in that person? No, because in an untreated type 1 diabetic, where is the sugar? No, it's not even in the cytoplasm. It's outside the cell, right? And these are all intracellular processes. So what would happen, for instance, now I'm an untreated diabetic and I just ate sugar, right? I want you to imagine what's happening in my cardiac muscle cell. In my cardiac muscle cell, I just ate sugar. I can't get it in because sugar enters most of your cells by the process of facilitated diffusion, requires a carrier. And in order to turn the carrier on in most of your cells, you need what? It's insulin. So why did I pick cardiac muscle cell? because cardiac muscle cell, if you do not feed it something that it can make ATP out of, it will die like every other cell, right? Won't be able to maintain homeostasis. And cardiac muscle tissue is amitotic. It cannot go through cell division, right? Doesn't even have any centrioles any longer. So with an untreated type 1 diabetic, of course, you're damaging all tissues, but which tissues are really important not to damage? Nervous system, cardiovascular, right? Does that make sense? So this will reduce the ability of those cells and all cells to maintain homeostasis. Can you maintain homeostasis in the absence of ATP? No, you can't. The cell will become disorganized. Does that make sense? Okay, so glucose enters cells usually by the process of facilitated diffusion in the presence of insulin. And then, of course, glucose is going to be broken down. We already know. And this is a catabolic or anabolic process, this. That's a catabolic process, right? It's broken down, and if you have oxygen, it'll be broken all the way down to get a decent amount of ATP. But now, let's go back and talk about glucose. Glucose is like a really, really useful sugar because every damn cell in your body can burn glucose, right? There are some cells that are really specific about what they can burn, but every cell in your body, including, importantly, your brain, can burn glucose and really likes to burn glucose. That means that glucose is the body's equivalent of money, yes? So, does it surprise you that whenever I have excess glucose, I have a mechanism of storing it so that when I don't have enough glucose, I have a method of retrieving it, right? Because it's money. So, if you got extra money, what do you do with it? You don't drop it on the floor, you put it in a savings account, right? So, let's talk about the glucose savings mechanism and see if you guys can think about some complex physiology. So, I'm going to give you some terminology. The terminology is potentially confusing because it's all really similar, but if you look at the roots, you'll be able to tell it from each other. So this is also right here, but I'm going to recreate this on the board. Ready? Okay, so first let's start with the term that you've already met, which is the breakdown of glucose starts with glycolysis. I mean, you have to do more than glycolysis to get it all the way broken down, but what glucose does through glycolysis is ultimately get broken down to ATP, right? Does that make sense? So glycolysis. Now, I'm oversimplifying, but I'm just putting this word here because we're about to do every word that starts with glyco, and I want you to be able to compare and contrast them, right? Now, of course, glycolysis is only the first step of getting all of the ATP out of it. Keep glycolysis in your head. Glycolysis will break sugars down ultimately to get ATP out of them. Yes? You with me? Now, what would you do? What would you want to do in times in which you had excess glucose in the system in a healthy person? You would want to store it, yes? Okay? Because it's money. So the process of, after I've used all the glucose I needed for my cellular processes, I can store the excess. And I can store the excess as glycogen, which is how animals store glucose. Does anybody know the primary organ that you use to store glycogen? Yeah, it's primarily, but not exclusively, the liver. You can store it a few more places. And the process, guys, of storing glucose as glycogen is called, look at this, glycogenesis, the beginning of glycogen. Does that make sense? Okay. Glycogenesis. Okay. Savings account doesn't do you a damn bit of good if you can't withdraw from it when you need it. Yes? So, obviously, this process must be reversible. Good? And the other part, look at the name for it. If you break down glycogen back into glucose so that you can use it to get ATP, this is called Glycogenolysis. So look at the name. Lysis is breakdown of what? Glycogen. And it's actually called glycogenolysis. I just said glycogenolysis. Okay, so let's look at these before we add the last term. First, compare these two. People get these two confused for obvious reasons. They both have lysis in it, right? But this one is the breakdown of sugars. This one is the breakdown of glycogen. Does that make sense? Okay, now, Let's talk about this for just a minute. Okay, so when would you do glycogenesis? When you had high or low levels of glucose? Glycogenesis, high, yes. I used what I needed and I still had excess, I would store it in my liver, yeah? When would I do glycogenolysis? When I had low levels. But do you think I can ever run out of stored glycogen? Yeah, you totally can, yeah. So if you're in starvation conditions or if you're in a situation in which you're so ill that you can't eat, you will use up your glycogen stores. How much do you have? It really depends on your own metabolism. And for instance, you guys know that like marathon runners will carb load? What they're doing, among other things, is that they are making sure they store the maximum amount of sugar that they possibly can in their liver and other places. But are they eventually going to run out of it? Yeah, they're eventually going to run out of it. So what happens in a situation, let's say you've got somebody with the flu and they just can't eat a damn thing for like 3 days, right? So after you run out of your glycogen stores and realize that your brain almost exclusively uses glucose for its nutrients, what the hell are you going to do? Is your brain just going to say, well, it was a good life, we had a good run, it's over? But it's going to find it wherever it can, but it still kind of depends on glucose more than other things. And so now what's going to happen is run out of my stores. So I ran out of my circulating glucose, right? And then I ran out of my stored glucose. So plan A would be to use what's circulating. Plan B would be to use what's stored. Plan C is, in a pinch, I can actually start making glucose out of amino acids. Where do you think I get the amino acids? Yeah, you start eating yourself, basically. You're going to start eating your own muscle first. If you start running out of that, you'll start eating your bones as well. Okay, there's proteins in your bones. This process is definitely not plan A, and it's called gluconeogenesis. Look at that name. The beginning of new glucose. Yeah? Does that make sense? Gluconeogenesis. Everybody good? So go ahead.
So it uses muscle tissue?
Muscle tissue first, and then starts using other structural proteins. It'll start breaking down your bone mass as well.
So it doesn't use fat in your body?
This is glucose dependent. Now, I'm not saying that when you're in starvation conditions, other tissues aren't burning fat, but your brain can't really burn fat very well. And so regardless of the fact that you are burning, for instance, the fat on your right thigh to keep other tissues alive, some tissues are glucose dependent, so they can't burn fat. Does that make sense? Okay, go ahead.
So is that why your muscles will atrophy?
Yes, your muscles will atrophy when you get into those situations. Yeah? So everybody understand the big picture? So I'm going to ask you guys a couple of logic questions and see if you guys can figure this out. This isn't stuff that you probably have learned before, but you can work through it mentally. Okay. So this is really glucose dependent. And of course, what she just asked, that's Alice. Alice just asked, we are going to, right after we finish talking about burning carbs, we're going to talk about burning fats. We're going to talk about burning proteins. But for right now, we're carb dependent. Okay? And there are tissues that are carb dependent. All right. So you guys initiating this process and this process and this process, all three of these are hormonally controlled. Okay, so stick with me for a second. And see if you can figure out which of those three processes would probably be initiated by insulin. So we know that insulin primarily does what to your blood sugar levels? Decreases them. And what it does first, of course, before it does any of this, is puts the sugar into your cells. Yes? After that, which of these three processes would you imagine? Not glycogenolysis, glycogenesis, because that's storage, right? Yeah? So insulin triggers the storage of the excess sugar into the liver. Okay? Now I want you to think about the diabetic for just a second and see that you got a double whammy. Do you see that you are not only having trouble with bringing the sugar into your cells when it's available, but also with storing it, so that when it's not available, do you have much to release? No. You have trouble with insulin. You have trouble with glucose absorption into the cells. You also have trouble with glucose storage. So can you see that that would make the booms and crashes of sugar and an untreated diabetic even more dramatic than you would have first predicted? Yeah, because we can weather it, because what happens in a healthy person without diabetes is that when your glucose level is what, high or low, you will do glycogenolysis. Which one? Low. You will do glycogenolysis. There's another hormone that is responsible for triggering glycogenolysis. And you might remember it because we talked about it before, even though we didn't use this term. What's the hormone that you need when your glucose is gone? Glucagon. Yeah, glucagon. So you have experienced glucagon before, right? You've gotten so busy that you didn't eat, and you're so freaking hungry that you're like hangry and stupid, right? You can't work a doorknob and everything's pissing you off, right? You're like, look, just look at you sitting there being a toddler. I hate toddlers. And then you eat something, and you're like, I totally love you. So what happens is you go through that period, and then you're like, I am going to die, and I hate everything. But then all of a sudden you're okay. And you didn't eat anything? You're like, oh, I can do this. That's glycogen release. That's glycogenolysis. Just got dumped into your bloodstream, and then your brain is like, I can do this for a little longer. So glycogenolysis is triggered by glucagon. Yes? And then it'll dump that stored glucose into your bloodstream so that you can keep going for a little while. But then you're going to run out if you don't eat, right? You're going to run out. And so you guys tell me, which of the hormones, insulin or glucagon, do you think triggers this? Gluconeogenesis. It's glucagon, but not first, right? If glucagon triggered anything first, it would trigger glycogenolysis first, right? And then what would happen once you ran out? Then gluconeogenesis. Yeah? Does that make sense? So that's sort of the physiology that's involved with it.
Say that one time.
I don't know what I said, but I'll say something. I never, whenever people say that, can you say that again? I'm like, I don't know what I said. At least you know I don't memorize my lectures. You just read off them. I have no idea. So glucagon, when your glucose is low, glucagon will trigger glycogenolysis first to release your stored glucose. Then after you run out of that, second will be gluconeogenesis. Do you think gluconeogenesis happens necessarily over the course of most days? No, but it certainly can. And it's necessary during certain periods to keep you alive. Like when you have the 72-hour flu, you can't eat a damn thing. How do you stay alive? Gluconeogenesis. First trimester of pregnancy, for any of us who've been through that, sometimes you can't eat a damn thing. You just can't. You're going to throw up everything. It's going to come out the other end. How do you stay alive and also keep the baby alive? Gluconeogenesis. That's how you keep alive. Among other things, you're burning fat other places as well, too. Does that make sense? Okay, so these terms, can you see that they are going to end up being relevant all semester long? Like, we're going to talk about them again when we get to the endocrine system. We're going to talk about them again in other body systems, right? So they're going to be important to you. And notice that if you pay attention to the roots, you don't have to do much memorization, right? Because the roots actually make sense, right? Okay, so that is carbohydrate and glucose storage and use. And now we're going to get into lipids in just a second. But please note that you will, and it's on the Melanie B page. I'll put it on the recording so that the other people can look at it too, because I forgot to mention it in the first class, but it's not due for a long, long time anyway. Okay, so let me show you. Just a second. Go ahead.
Maybe a weird question, but how would the reflex arc for this work? For example, I understand how it works from when there's low blood sugar, sugars like glucagon from the pancreas, but when that negative error signal we're out.
Yeah, that's actually a really good question. It's just a reflex arc that uses the level of glucose as the indicator of when to start and when to stop releasing both insulin and glucagon. Does that make sense? So the new level of, so if, for instance, I was really low in glucose, yeah, then my pancreas would say that error needs the effector response of releasing glucagon. You with me? Glucagon would go to the effector, which is the liver. Yeah? The liver would release glucose into the bloodstream. That new level of glucose would feed back to the pancreas, and as soon as it got into an acceptable level, it would inhibit the release of glucagon. If it got above that, it would stimulate the release of insulin. So it's actually a really good question. It's a little reflex arc that just involves hormones in the communication process, right? And if it worked properly, if this works really well, and your acceptable glucose range was here, can you see that it would just do this? Yeah? I mean, maybe if you went to swig, it'd go like that, but that shit's really bad for you, you know that? It would go like that, but then you would bring it back down. Does that make sense? And it's just using the new input as the new level of glucose. That's the new thing that is being sensed. That's the new variable that's being measured. Does that make sense? Almost?
I guess I'm just trying to figure out how the gluconeogenesis.
Oh, it would have to be low levels for a very long time, and then you would have had to use up your stores. Yeah.
So there would be like an error from the liver saying, like, hey.
An error from the pancreas, an error from the liver, and then you're like, I tried to use this effector, that effector failed. Then you go to the next effector.
So there's like multiple sensors.
Yeah, multiple sensors and multiple acceptable ranges.
Which is
Yeah.
the hard part to it.
It is.
You just learn like, like one sensor.
Yeah.
It's like, no, there's like this many.
Yeah, it's really by any means necessary, right? If you realize how important the outcome is, right? So then what are you going to do? You'll do the one that is cheapest and fastest first, and then if that fails, you're going to do something that's far more costly, but still keeps you alive, because the outcome of all of this is maintaining homeostasis, right? It's staying alive. Okay, everybody good? Yes, phoning a friend, phoning your dad, right? You're like, I tried everything. I don't know how to change this damn tire. Dad, no. I do know how to change a tire, and I'm the one they call now. But I got them all AAA on there. I'm like, you can call them. I pay for that crap. Okay, so let me show you something that is an assignment for this semester. It is not due until you take your final, but and it's really relatively easy. So I'm just going to show it to you today for the first time. It's on melanieb.com. Oops, and it is just a Word document, and all it is the liver is so damn important that I want you guys to keep a list of liver functions that you will turn in to me with your final, and you will organize the activity, the purpose, and the body system that it has to do with. Sometimes it's more than one body system. And to give you some encouragement to do that, it's worth a 5% bump on your final. If you do a good job. If you do a crappy job, I'll give you 1%. Okay, so I know all of the times that I talk about liver, both in lecture and lab, all semester long, so I know about how long this should be. Notice also that if you are a smart person, you can also do a search of all of your PDF documents for the word liver. Yes? And then talk about it. We've talked about like 4 today already, and we're about to talk about some more. So this is just a really easy way of like giving you guys sort of a global appreciation of all of the crap that the liver does. Like, you are seriously, seriously dependent on your liver, and you're about to see some other ways. So you can see now that, of course, it stores and releases glycogen. Okay, so, and you can just work through that throughout the semester and keep it, and then like get it organized. Okay, so now, what we were talking about just then is exclusively carbohydrate metabolism. But of course, you've been a person for a while, and you know that you don't just live on carbohydrates. You also use lipids, yeah? And proteins. So let's talk about catabolic processes with both of those. These you are probably less familiar with, but I'm not making you memorize the chemical equation. I am actually making you look at how it's related to the stuff that we've already done. Okay, so when we are talking about lipids, what you are going to have to do is, there are big complicated lipids. Just like with carbs, you're going to have to break them down to something smaller that you could actually burn. You may or may not know the names of those smaller things, but let's just talk about this. Okay, first off, lipids are more energy dense than carbohydrates. which is great when you, for instance, need to eat something that's energy dense that'll last for a while. Lipids are good. It's not so great when you're trying to burn them off of your body because you actually have to work a little harder to do that. They're more energy dense, and I think it's like 9 versus 12 grams per kilocalorie or something, or kilocalories per gram. The liver is intimately related to anything related to lipids as well, 2 ways. First, when you take in lipids, you will not be able to absorb them without bile salts made from the liver. So that's the first thing, okay? And then second, bile salts do breakdown so that you can do absorption. And then second, the liver makes proteins that are necessary for lipid transport in the bloodstream. So if basically here, what I'm saying is if you have liver problems, you have lipid problems. It's going to cause problems. Okay, so the process of lipid digestion or lipid breakdown is called lipolysis, not glycolysis. Not surprising, right? Lipolysis. So let's just go through the big picture of what you do with lipid breakdown. First off, bile salts that are made from the liver get dumped gallbladder, right? And then store them in the gallbladder, dump them into the intestine, right? If you don't have that, then you're probably not going to be able to break down large lipids to smaller lipids for absorption and also moving into the cells, okay? Got it? So here's the basic breakdown. You may not have heard of this before. You take a big lipid and you break it down via lipase from the pancreas and also bile salts from the liver to 2 things, fatty acids and glycerol. So let's look right here. Do you see glycerol and fatty acids right there? Okay. These are the things that you can burn. Okay. Anything bigger than that, you break down to fatty acids and glycerol. Okay. Now, please note that even though you're not doing memorization, I do need you to get the big picture. Notice that fatty acids are not glyco anything. They skip glycolysis altogether, and they go in either right before or right after Krebs. You with me? Okay. Which means what? Do they require oxygen or not to break them down? Yeah, they do. They absolutely do. Everything that happens in the mitochondria requires oxygen, so you need oxygen for breakdown of fats. Now, there is glycerol, which is like this fat-sugar combo. That one actually goes into glycolysis, but break it down mostly to fatty acids. Everybody okay with that? Okay, so am I talking right here about anabolic or catabolic processes with fats here? These are catabolic processes, right? I'm breaking them down. I'm trying to get ATP out of them. They will generate more ATP per gram than carbs, but it's a similar process that we're using there. Okay, so fatty acids will enter metabolism after. Okay, so I'll talk about one thing and then I'll talk about ketones real quick. Okay, so now here's something interesting that is relevant to all of us. What is relevant to all of us is that you can actually eat too many carbs and end up doing lipogenesis. Can you get fat from eating too many carbs? Can you get fat from eating too much fat? Yeah. Can you get fat from eating too many proteins? Why? How the hell does that work? It works because one of these reactions is reversible. Do you see that no matter how you get this acetyl-CoA, you can make it into fatty acids? Yeah? Whether it came from carbs, whether it came from proteins, whether it came from fats, you can make it into fatty acids. So when, for instance, you stored all of the glucose that you could, and you still had extra carbs left because you went to swig twice in a week. Okay? What you going to do with it? You're like, I already stored all of my glycogen that I could possibly store. And now what am I going to do? I'm going to give myself a gut. That's what I'm going to do. I'm going to store it as fatty acids. I'm going to store it in my adipocytes. Adipocytes? Yeah? And they store lipids in your adipose tissue. See if it surprises you even at all that one of the major triggers for lipogenesis, basically gaining weight, is one of these hormones that we talked about. Which one makes sense? So, insulin did what? It made your cells take up the carbs, yes? Then it stored all of the excess carbs that it could in the liver. Then I ran out of storage space. Guess what it triggers? Nope, it triggers lipogenesis with the rest. Yes? One of the many, many reasons why eating carbs in combination with either other carbs, with fat, with any other food, is more likely to make you gain weight than not eating very many carbs, is because one of the major triggers for lipogenesis is insulin. Does that make sense to you guys? Do you see that what this did? Do you see? What you did is insulin did what? So insulin is first, plan A, cells take it in, yes? Plan B, I've still got excess sugar, yes? Plan B is stored in my liver. I've still got excess sugar. What do? I store it in my fat. Yeah? Okay. So one of the things that they figured out, and they did not know this until about the last decade, they did not know that one of the major triggers for weight gain is insulin, right? Which is how you get like metabolic syndrome, which leads to type 2 diabetes, which is usually associated with obesity with most people, right? Unless you have a really, really fast metabolism. Do you see that what you're doing when you take in carbs is storing, and then storing the excess as fat? Does that make sense? Okay. It's also one of the reasons why when someone is diagnosed with type 1 diabetes, one of the first things that happens other than peeing like a racehorse is that you drop like tons of weight. Why? You're losing your trigger for lipogenesis. Does that make sense? Do you guys get what this is talking about? You're not nodding very much. Who has questions? Who has questions?
Can you repeat that last part about if
Yeah, just
somebody
the last
loses the first sign is I'm losing weight?
Oh, no, I'm not talking about type 2 diabetes. I'm talking about type 1 diabetes, which is the inability to make adequate levels of insulin. Okay?
Oh,
So-
that's storing.
So you can't store in your liver, you also can't store in your fat, regardless of the amount that you're eating. Your lipogenesis is actually inhibited. So a lot of times what happens when people are first diagnosed with type 1 diabetes, type 1, not type 2, is that with type 1 diabetes, you don't have enough insulin. So they're going to pee like crazy for reasons that will become obvious later. But a lot of times also within the course of a month, type 1 diabetes is autoimmune. Yeah, a genetic predisposition, but it's autoimmune. Whatever destroyed your pancreatic islets, and you now no longer make adequate levels of insulin, and so you can't really store anything. You just drop weight like crazy. Like you'll lose like 15 or 20 pounds over the course of a month, a lot of times. Does that make sense? Now, type 2 diabetes is exactly the opposite problem. Type 2 diabetes is too much insulin over long periods of time. So you generally have a lot of glycogenesis with that. Is that okay? Okay, you guys all right with that? You sure? Okay, so the thing that I wanted to tell you is the thing that I wish I had known when I was young, before I decided that carbs were among my favorite things in the history of the world. And they still are, right? I love them so much. We have an intimate relationship. Anything complex. I don't really like sweets, but God, I love bread. Bread's amazing. I wish I had known that. And I'm trying to train my children out of like depending on carbs as the basis of their diet, so that when they get into their 30s and 40s, they don't have to start thinking about this quite as much as I did. Okay, so the other thing is, let me talk briefly about the ketogenic diet and what it has to do with this story and what people think about the ketogenic diet. You've heard of the ketogenic diet, right? This is the one that's super duper popular right now. The ketogenic diet also has to do with this process, because I want you to look. So with the ketogenic diet, what you do is you start changing your dietary patterns, and you do it really dramatically at first. Primarily what you do is really dramatically reduce carbohydrate intake, dramatically. But at the same time, you also massively increase fat intake and increase protein intake, not as much as fat though. Okay, so what you're doing here is, of course, you are going to reduce the insulin released, yes? Okay. And reduce lipogenesis by doing that. And a lot of people do lose weight on the ketogenic diet. Is it without consequence? It is not without consequence, because here's something that happens when you do that. When you, for instance, primarily use fatty acids as your source for catabolic processes, you produce this byproduct called ketones. The reason that you can live with really, really low carbs is because the brain can burn ketones as well as glucose. It doesn't like to. It gets super pissed off at first when you try to make it do that. It's a difficult shift to do. But one of the reasons that this is potentially problematic and should not be undertaken lightly is that ketones are acidic, and they will change the pH of your bloodstream. And you might be able to metabolically handle that, but she might not. It depends on the person, right? So they change your pH. And the other thing is that when people do the ketogenic diet, they are reducing carbs, yes. They are increasing fat. They don't increase fat with just good fats. They increase fat with animal fats. They eat an awful lot of bacon and eggs and cream and milk. And this causes a potential problem with your cholesterol level. Now, strangely, not everyone who does the ketogenic diet gets high cholesterol, but some people do. Interestingly, my husband and I tried it over the summer, and we both did it kind of experimentally, and we actually told our physicians that we were going to do it. And then we went and had our blood drawn after 2 months of doing the ketogenic diet. My cholesterol level was fine. His was through the roof, just bonkers through the roof. And we eat around the same, we ate the same thing. He eats more, he's huge. So your metabolism might be able to handle it without having a massive spike in cholesterol, but I'll tell you that cardiologists do not love this diet. They do not love it. This diet, interestingly, the ketogenic diet, was developed as a medical intervention. It wasn't developed as like a fad diet. It was developed it was developed for epilepsy. Yeah. So this one's really interesting, and it's still used for epilepsy, FYI. I educated myself quite a bit more about epilepsy last semester because I had a student last semester who had epilepsy, still does, and she's in my class this semester as well. So I read a bunch and was trying to figure out the physiology. So they use this really high-fat, really low-carb diet to manage seizures in people with epilepsy. It's a difficult diet to stick to. It really is. It works really, really well in people with epilepsy, but they have no idea why. They do not understand the physiology. Some research papers propose that it has to do with changing the pH. Some of them propose that it has to do with changing the hormonal environment. But if you can really, really adhere to it massively, it reduces seizure frequency in most people with epilepsy really effectively. Sometimes to the point that they don't have to be on medication. But we don't know. We don't know actually what it's doing. It's also got potential neurological benefits for other things as well. But it is not easy, and it is not without its potential costs, just like every other diet. This one's a really extreme diet. It really is. So that's the ketogenic diet. Ketones are acidic, and they're produced from excessive triglyceride breakdown. And they can be used by the CNS as partial alternative to glucose, but alter the body's pH with ketoacidosis. You have to be super duper careful about this diet if you have other metabolic problems with like diabetes. So it's not just that you can do it to reduce your carbs. So anyway, that's worth talking about. What? Yeah, you can get fatty liver from it too, but not everybody does. Yeah.
Like I know a lot of people who do the ketone diet.
Mm-hmm.
They always smell kind of like off.
They what?
They smell kind of off. Like,
yeah. Oh, it changes. Yeah. So both Atkins, which is a low carb but not as high fat, and the ketogenic diet, the generation of ketones in your bloodstream changes the chemicals that are released in your sweat and in your breath. It usually causes sort of a chemical smell. In your breath, and that's a lot of times what diabetics smell like as well if they're not being treated properly. So I'm not saying don't do this. I'm saying that not everyone can or should do this. Some people are really healthy when they do that. It is something that you would probably want to go to the doctor after you had tried for a little while to make sure that you are not affecting your long-term cardiovascular health. Go ahead.
How does this affect your gallbladder? My boss tried it and he had a gallbladder attack.
Oh, interesting. Well, all of those fats, most of us won't, but if he had a predisposition to gallbladder problems, that's a good way to get one. You might not know that you had a predisposition to that. What's going to happen is you're going to need a whole lot more bile salts to absorb all of the fats that you're taking in. And if your gallbladder cannot keep up, it's going to actually cause a gallbladder attack. But that wouldn't happen to most of us. But basically, when you're doing something that extreme to your system, you're doing an experiment on your system. And a good scientist, when they're doing an experiment, they realize that they should probably track the outcome in some way or another, and they should hold all of the other variables constant, right? Okay, and it's difficult to do. In addition, if you are doing the ketogenic diet without doing exercise, it tends to be harder on your system than if you're doing it with exercise. Exercise tends to clear some of the other stuff out of your system. So anyway, that's the ketogenic diet and the relationship between insulin and fat storage, which is always really good to tell people. Okay, the last thing is, let's talk about cholesterol. Even though it's not directly related to what we're talking about here, let's talk about cholesterols and what they have to do with the whole lipid thing. So when you are, cholesterols are from animal products, yes? Plant products have damn near zero appreciable cholesterol in them. All animal cells, including yours, have cholesterol in their cell membrane, okay? Now, cholesterol is not one thing. Cholesterol is, it should be separated into at least two categories, because there is your good cholesterol and there's your bad cholesterol. The good are the high ones, HDL, high density lipoprotein. The good cholesterol you get primarily, you can get a little bit from your diet, but you get it primarily from exercise. Okay? The only appreciable way to really increase your HDL is from exercise, and that's your good cholesterol. And the good cholesterol is good because it makes the bad cholesterol less likely to stick to your blood vessels. You with me? Okay, so LDL and VLDL are low-density lipoprotein and very low-density lipoprotein, and those are the bad cholesterols. And under most circumstances, you get those from eating animal products. Even if you're a vegetarian, you can have high cholesterol. If you're a vegan, you're going to have a hell of a time getting high cholesterol. But eggs, right, cheese, dairy products, those have cholesterol. And of course, bacon has boatloads. Any fatty meat has tons and tons of cholesterol in it. So the problem with just talking about cholesterol as one number is what you really need is the ratio of your H to your L's. So you don't just need your cholesterol level, you need your cholesterol ratio, right? Because can a person who is really, really physically active get away with actually eating far more cholesterol in their diet without affecting their cardiovascular health? Yes. You can't get away with it forever. And eventually you're not going to be very active, right? And if you still have that diet, you're going to be screwed. So that's the problem, right? You form that diet when you're active, but then do you, when you're 80, all of a sudden start eating, like all plant-based? No, you don't change a damn thing when you're 80. You're like, I'm 80, screw you. The cholesterols, the problem with cholesterol, and you know this, and I'm oversimplifying, but just let's make sure that we're all on the same page. The problem with your bad cholesterols is that they stick inside the walls of your blood vessels, which will, of course, eventually increase your blood pressure. And you have to compensate for the increased blood pressure by your heart working harder, right? So basically what you start is this slow upsweep that leads to heart disease, right? But the problem, and this is the issue, is that it starts before you have symptoms. You don't have angina, you don't have high blood pressure, you just go to the doctor and they say your cholesterol is high, but you feel fine. So what do people generally do? Not a damn thing. They just keep doing what they were going to do. But this is the predictor of the blood pressure that is coming and then the heart disease that is coming after that, especially if you have a genetic predisposition to this. So the problem is that the warning signals, although the doctor can hear them and they can tell you about them, what I think is, yet again, we should have been educated about physiology long before college, right? You should have said, the reason that you want to watch these foods is this, the reason you want to watch these foods is this, the reason you want to put exercise as a part of your habit is this. And then at least if you knew you weren't going to do it, you knew the long-term outcome, right? And maybe you would have started it in your 20s instead of in your 30s or your 40s. So your cholesterols here are primarily related to your diet and exercise patterns, is what they're related to. Americans have a ridiculously high predisposition to high cholesterol because we tend to be relatively inactive and eat lots and lots of animal products compared to the population of the world at large. There are some drugs. So generally what people do is when the doctor says you need to reduce your cholesterol level and increase your exercise, they say, give me a pill. Guess how well pills work, that kind of intervention, compared to your normal homeostatic control mechanisms for changing. They don't work very well. I mean, they don't not work, but they don't work as well as your homeostatic control systems, right? So I am trying very, very hard to teach my kids this stuff early. They don't ever want to listen, but I'm hoping it just gets in there like a little irritating earworm, right? I'm like, you stop. Go ahead.
Hey, I have a question on this.
Okay.